Welcome everyone to the monthly CHIBI faculty seminar series. Today's speaker is Aristotelis Tsirigos, and he has a PhD in computer science from NYU, and his research focuses on epigenetics. Dadu, welcome.
Thank you, Armina. Hello, everybody. So some of you have probably seen parts of that before. There is some new figures that you may be interested in. I'm counting on everybody to ask questions because I feel very sleepy today. I didn't sleep well last night. And I'm looking at Adriana because she always asks questions, and that's always nice. So my talk is going to be about characterizing aberrant histone modification patterns in T-cell leukemia. And this is work with Dr. Ifandis' lab, and specifically Panos Diachristos. So the goal of the study was to study histone modifications in cancer progression, and specifically, as I said before, in T-cell leukemia. Dr. Ifandis' lab has a mouse model that recapitulates very well the main characteristics of the disease. And also we looked at patient data and cell lines where we also did experiments. And our tools were genomics as well, so RNA sequencing, chip sequencing, and computational analysis. So in this disease, I'm wondering if there is a way to move slides to, do I have to hit here? That's the only way? Yeah, okay. So, okay, a few things about the disease itself. So Notch is a major oncogene in this disease. More than 50% of the patients have a mutation in this oncogene. And I'm going to show something about the pathway, Notch pathway. So Notch has an extracellular domain right here, gets activated. Upon activation, there's cleavage by gamma secretase, and then the intracellular part of Notch enters the nucleus and it activates the oncogenic program. So one of the major tools we have is gamma secretase inhibitor that blocks detachment of intracellular Notch, and then we can block the disease in cell lines. As I said before, we also have, Dr. Ifandis' lab has an inducible mouse model where the intracellular part of Notch is inserted and is activated upon injection with tamoxifen, and then you can actually see disease progression, and I think within a few weeks, these mice get leukemia. So our very basic question that was almost 4 years ago, was whether, at least in the mouse model, you have anything that goes wrong in the epigenetic state of the T cells when the mice get leukemia. That's a very broad hypothesis, of course, and we wanted to use a data-driven approach to really try to refine it, to see what exactly does it mean, what kind of epigenetic state differences you may have between leukemia and normal T cells. I'm assuming all of you know the basics of epigenetics and histone modifications. I'm not going to be talking at all about DNA methylation. This is only going to be about histone modifications. And in this study, we looked at specific activating marks. I think that's what you have to remember throughout the talk, that we had some activating histone modification marks. So when you see them, it means that the associated genes are activated. This is K9 acetylation and K4 trimethylation. And we also have a repressive mark, K27 trimethylation. So genes that, let's say, possess this modification in TSSs, usually they're repressed. And we did Chip-seq for Notch as well, and expression at that time it was microarrays. Okay. So the goal was again to find some interesting changes, statistically at least, to find changes between leukemic and normal T cells in mice. And the key ideas of the analysis was to do a very comprehensive data set quality control, to to understand our data. Again, that whole study started 5 years ago. Chip-seq was not very much used at that point, so we really wanted to understand how it works, the properties of the data. Then we wanted to do a multimodal dataset validation with other published data and datasets. And after doing all these, we were planning to proceed to find epigenetic changes, as I said before, between leukemic and normal T cells. And the real goal was to refine the original hypothesis. That was a hypothesis too. Maybe we wouldn't have found anything. So the first data set we got was a very simple microarray data set between normal and leukemic double positive cells. And we just verified that classic notch targets like HES1, delta-X were actually upregulated in leukemia compared to normal. Then our task was to analyze the CHIP sequencing data. So first you look at the actual reads that come out of the Illumina machine. You have to remove, these are kind of standard now, remove standard PCR duplicates, ignore repetitive loci. In fact, we did some analysis to see if that affects our results at all. We didn't find any evidence of that at that point, even with shorter reads at that point. Of course, do false discovery rate when it comes to peak discovery, check reproducibility of the results. And then the real important task was to find what we call differential peaks. So when you do the Chip-seq analysis, normally you call peaks. In our case, we had leukemic cells and normal cells. We really wanted to find statistically significant differences between the two states, different peaks for all these Chip-seq data sets we had. The very first analysis was just to see the reproducibility of the replicates we had. So we did everything in duplicates, and there is a nice correlation you see here, both in the normal double positive cells and the leukemic ones. You can clearly see here that the repressive mark is not correlated at all with the activating marks. Whereas the activating marks you usually see together in TSSs. So the next step in our quality control pipeline was to integrate, to do a first pass integration of this data with the expression data. So we separated our genes in highly expressed and genes of low expression. So you see the green is high, red is low. And then we asked the question whether highly expressed genes really possessed or not these marks one by one, both in double positive cells and leukemic double positive cells. So what you see here is a panel. You can see the repressive mark first, both in normal and leukemia, and then the activating marks. And as expected, you see the difference. You see that in activating marks, highly expressed genes actually show this nice pattern in their TSSs, whereas genes of low expression don't. Same here. And you get the totally opposite, you have the totally opposite effect here. You have repressive mark in repressed genes, as expected. So you may think this is just boring, and it usually is and usually has to be, because you have to get what you expect. But even when you do this boring thing, trust me, I don't want to put on boring slides, but this is not, this ended up not being so boring after all, because you see that even when you do this analysis and you get almost what you expect, in leukemic cells you get this difference. So yes, in lowly expressed genes or highly expressed genes, you get this, what we call a valley. So there seems to be some defect here already. We don't know what it is, but it could be something interesting. So then another kind of analysis you can do to extend what we did previously, these are ROC curves. You can see how many of these expressed genes you can recover by just looking at histone modifications. So by just looking at activating marks, can you really capture all your activated genes? And you do pretty well. You go very quickly to, I would say, over 80% with a very small false positive rate for both marks. K9 seems to be a bit more specific. And then also interestingly here, and maybe that's another hint of something is let's see, I'm sorry, here. These are kind of not much, right? Okay. So you can see here, this is now the opposite. For K27 trimethylation, this is the repressive mark. The question is whether you can capture the repressed genes. And we're doing good, but not as well as we did with the activating marks. And that's normal, I would say, because there are other repressive marks as well. So it's not just K27, you have K9 trimethylation, you have DNA methylation. So it's not surprising that you cannot capture 100 or 80 or 90% of the repressed genes just by using this mark. And these are, I'm sorry about this, these are not aligned, so this should be here. This is now in leukemia, same thing, same thing. Okay, so how did we solve this problem of identifying epigenetic changes? So we came up with a very simple method to identify enriched genomic windows. This is something like peak calling, but we also compared to randomized controls, both locally and globally. Then one interesting, I think, step, and I can show you some data on this one. One interesting step was to do a quantile normalization across all the windows, enriched or not. Usually what is done is you normalize by the total number of reads. That's the standard thing you do. We found that quantile normalization can correct for a few things. So after that, we removed outliers, we computed fault changes, and then we computed a false discovery rate on those fault changes. Another interesting little thing we did here, and I'm going to get into some detail, is that we did it by intensity level. So I'm going to show you some figures, but I think it's clear that when you have a higher, stronger signal, then your false discovery rate, when you compute the fault chain, is going to be smaller. But if you go to lower and lower and lower intensities, meaning fewer reads, your false discovery rate can go up. So, yes? Yep.
So you first do the fold change, some kind of cutoff, and then you do the false discovery rate on those?
There's no cutoff. You compute all fold changes.
You do
all fold changes, and then basically I'm going to get into details. Okay. You compute the distribution of observed fold changes to some randomized control, or not necessarily randomized, as you can see. So this is how the replicates look like. K27, replicate 1, replicate 2. This is all in duplicates. So going back to quantile normalization, we noticed that sometimes the replicates are good. So this is perfect. They follow, let's say, a straight line. And again, as you get lower intensity, fewer reads, you see the spread. So it makes sense that when you move to lower intensities, you need to be extra conservative to call differences. So most of them looked fine, but this one and that one, you can see that they're not completely straight lines. Yet again, there is some signal here, but if you consider this to be your model, then it's clearly wrong. And quantile normalization fixed that. Okay, so this is a figure of how of the different steps of the analysis. This is one condition, this is the other condition. So let's say tumor and normal, and these are the two replicates. And again, you remove outliers, so whatever doesn't fit your model, you just remove it, and then you proceed with what's left. Of course, this is after quantile normalization, and then you call the differences. But as you can see, as you move down in intensity, your cutoff, so whatever I'm marking in different colors is significant, 5% false discovery rate, but it's not a straight line, because as you move down, you have this effect of using a different cutoff, I guess, for calling it significant. So we don't use the same fold change cutoff at every level. We change it. The other thing we do, so I mentioned before, we computed all fold changes, and we wanted to compare it to some other distribution. So we found that a good distribution is to compare within replicates. So when you compute the fold changes between the two states, but for our background distribution, we used one replicate against the other. Right? So one replicate against the other shouldn't give any fold change in theory, but of course it does. So this is our background. Okay, so some evidence about the potential effect of this quantile normalization was that, so when we got more data sets, the ENCODE projects, and that's really a few years after the project was done, so we did a classic PCA, and that's thanks to Igor's nice code. I don't know if he's here. But, okay, so we noticed that fine, if you just do a PCA on the original data, they cluster kind of okay. You see the repressive marks here, then the other type of repressive mark is here, and then pretty much every activating histone modification is clustered together here. But when you do quantile normalization, and this is just one cell line, and it's a cell line, and it's the ENCODE project, so it should be perfect. And still, I was not so satisfied with the other one, especially when I compared it to the quantile normalized version of the same plot. And I think that's pretty nice. Things are very well separated, and colors match, so same color for the same type of histone modification. This is the control. It's right in the middle, and the two types of repressive marks are clearly far away from each other. So I was really satisfied with that. And again, that was just one cell line. If you add a second cell line, then this is how it looks when you don't do a quantile normalization. I think it's pretty ugly. I mean, you can still argue, yes, colors are kind of close, but I think that's much better. Okay. So the separation and the clustering is much clearer, and you can even see. Unfortunately, we didn't have matched histone modifications between the two cell lines. Only a few of them were common. So this primarily Notch in CUTLLI, and DND, we don't have Notch. Chip-seq done by ENCODE. But you can see that although this comes from CUTLLI, a different cell line, it actually clusters very well with a K27 of DND41, the other cell line. Same thing kind of for K4 trimethylation, at least it's in that group. But this can also kind of highlight the differences between the cell lines. So they're not next to each other because they're different. But everything else is really not only clustered very closely together, but actually leaves some room for the other group to breathe. So I don't know which can comment. I think I lost my microphone here. Yes. Yes. So in the method I described, of course, just for the histone itself, for one type of histone. This one, everything normalized.
Why would you expect them to have the same distribution?
I didn't. I mean, when I did it, can I explain it fully? No. So it may just mean that the relative levels, so it's a very good question. It might mean, I can only speculate, and I would say it might mean that the relative levels are important. So if something has K9 trimethylation and it has a level of 10, it doesn't so much really matter, only compared to where it is in the distribution. What percentile? Maybe, I mean, that's just speculation, I don't know. Okay, so after we called these significant differences, we really, to trust them, because we had no idea what to expect, to trust them, we did a gene set enrichment analysis to see what genes they belong to and if that made sense for the system we were studying, and it actually did. Both for the activating marks, you can clearly see actually the specificity. We're talking about T cells, and you see T cells. That was pretty good. And that's changes. I have to say that's not trivial because you're looking at changes. You're not looking at absolute levels. That would have been kind of trivial because you can see immediately what's activated. Looking at RNA-seq, K4 trimethylation, you find the genes that are activated. But to see the differences, it's not so easy. And same thing for K27 trimethylation. You can actually see that these genes that lost K27 in leukemia, they're actually PRC2 targets, and PRC2 is a complex that methylates K27. Then you do a genome-wide characterization. That's probably really boring, so we skip that. And then you can see nicely in a heat map what kind of, let's say, more complex patterns you may get and how you can integrate it with differences in expression. So we really saw that genes that are upregulated in leukemia are mainly characterized by this loss. You can see it here. This is normal. This is leukemia of the repressive mark. So it seems, even just by looking at it, that it is the most important epigenetic change, at least within the changes we tested for, that explains best the differences in expression. And as a control, you have, we put the downregulated genes, and you can see that this is not true here. So this is not something trivial. Somebody might say that, okay, of course, you lose K27, then you have upregulation. But here in the downregulated genes, it's a different story. Downregulation cannot be explained by a change in K27. Instead, it can be explained by a change in the activating mark a little bit. You can probably not see it by eye, but when we do the statistics, we know that this is this change in this mark, the activating mark, can explain the downregulation to some extent. And there was a nice cluster in here with some very well-characterized notch targets that actually had both loss of K27 and increase in the activating mark here, actually in both. So that was kind of cool. And we got different patterns. We actually did a complete analysis of all the patterns, as I alluded to before, all the combinations in different regions of the genome, all the epigenetic events that we found, epigenetic differences we found using that method. We actually did all pairwise combinations and in triplicates, and we just ranked them by p-value. And as I said before, K27 trimethylation loss from normal to leukemia was the epigenetic change that best explained the data, at least the upregulated genes. This is just one example. You can even see it in the browser. You can see that HES1, a classic notch target, loses K27, leukemia and normal again. And there is some increase of K9 and K4 trimethylation. And same for delta-X and other target, exactly the same thing. And I would say, again, this is something I wouldn't say trivial, but you really need, you do your statistics, you do your global analysis, you find some signal, p-value 10 to the minus 10. But when you actually look at the browser, it's some very terrible differences that nobody would believe. You really have to go down and find good examples. It's not just the statistics that you have to do. And another type of analysis was notch-centric analysis to just show the connection to notch, the actual connection between loss of K27 and notch binding. So you can see that here we split the genes into bound by notch and not bound by notch. And that's what you see in normal cells, genes bound by notch in leukemia. Right? That's important. Let's not be confused here. So genes bound by notch in leukemia, and there's no notch, and Yanis is here to correct me if I'm wrong. So there's no notch in normal double positive cells, so they're not bound by notch. But what you see is not only they're not bound by notch, which we know, but they're repressed by K27. And these same genes seem to have this opening here. Not only they're bound by notch now, but they're bound, and K27 is kind of pushed to the sites.
So, how is that different? First of all, do you have difference in, let's say, the expression of the corresponding complexes that are required for the methylation? So, like
Expression levels?
yes.
No,
The methyl
I don't think so.
phosphorylases?
No.
No. So then, is it the nodes that does the job in this case? Does nodes bring?
Yeah, so Panos in the lab did some experiments to show that that NOTCH evicts EZH2. So that would be my. So we didn't. So just to answer your question from a bioinformatics perspective, we didn't do a ChIP-seq for EZH2, at least a satisfactory one. And these are some enrichments. Again, notch-centric analysis to show that notch co-localizes with loss of K27, not with gain. And it also, and more so than it does with K4 trimethylation gain. It does co-localize with this, which was kind of interesting. And we never fully explained. So if you have any ideas, because normally it should kind of co-localize with the opposite, gain of K27 trimethylation. But that was not the case. I had one theory but Yannis didn't like it, so we didn't put it in the paper. Of course, because you didn't like it. So, okay, so now we established that K27 loss, when you go to leukemia, is the most important, at least of the ones we tested, epigenetic changes, epigenetic change. And of course, we knew that PRC2 and EZH2 in particular methylates K27, so we wondered whether there is a defect in this gene, and luckily at that point, Dr. Ferrando's lab from Colombia had the data to support that. He had patient data where he showed that EZH2 is actually inactivated in 25% of patients. There's some deletions, some different events. So that was nice. And this is a summary of the different hypothesis we had. Notch antagonizes PRC2, evicts EZH2 as Harris asked, so we had data for that, experimental data for that. Panos did the experiments, of course, and some other experiments he did. Maybe it's not exactly just evicting EZH2, but maybe evicting the nucleosomes, so he had some data on that. And again, as I just mentioned, we found, Dr. Ferrando's lab found that 25% of patients had inactivating mutations in TALL, in EZH2, or other PRC2 members. So that concludes the first part of the talk. Do we know what time it is?
It's about 2 o'clock.
It's 2, okay. Okay, so.
Exactly 2 o'clock.
Great. So let's move to the second part. So after we completed this study, we wondered whether inactivation of EZH2, so we know that K27 loss is a major characteristic now, but we wonder whether EZH2 itself and the fact that it's inactivated in patients can fully explain what we see. And of course, that cannot be the case, because only if you see it from another point of view, only 25% of the patients had inactivating mutations, the others didn't. So is there something more, is there any additional information we can get from this K27 loss? Or is it just the whole story was just EZH2 being inactivated? And Panos had this great idea to look at the enzymes that do the exact opposite. So EZH2 methylates K27, which we see it's lost. Fine, because EZH2 is inactivated. But maybe in some patients, in some cases, it's not actually EZH2 that methylates and represses the genes, but it's the demethylases, UTX and JMJD3, that promote the oncogenic program. So the two demethylases, just a few words. UTX is constitutively expressed. That's the main thing we need to remember here, whereas Jumanji is induced upon inflammation. So based on that, Jumanji was more, I guess, likely candidate at that point. And to establish that, we first looked at patient data. So there was this nice data set, about 600 patients, was just published, microarray data. And what you can see here in different colors is different types of leukemia, T-cell leukemia, B-cell leukemia with different rearrangements. And what you see is that Jumanji in our type, in the type of leukemia we're studying, is the most highly expressed. So you can see that here. Additionally, it follows the pattern of Notch, the pattern of expression of Notch, with some exception here. So these are some quote-unquote normal T cells. I don't know exactly what happens here. I guess Notch is needed during development. That's right, I think. So I think that explains it. But you can see when you focus on leukemias, Notch again has the same pattern, highly expressed in T-cell leukemias compared to other types of leukemia. UTX, on the other hand, didn't have this pattern, so it was kind of flat. Then we continued with the analysis to just try to contrast all the kind of evidence we had for the two demethylases to really establish that they're different. And a very simple thing is just since we had 500-600 patients, we did a correlation analysis of UTX or Jumanji against every other gene, and then we kept the highly correlated genes, and we looked for enrichment in different gene sets. So you can clearly see that it's Jumanji and not UTX that actually correlates, Jumanji or UTX correlating genes that are enriched in K27 trimethylation or PRC2 targets or Notch targets. So I think from this, it is becoming clear that, let's say, potential Jumanji targets that are correlating in a very nice cohort of patients, these are the ones that are enriched in what makes sense for this type of K27, let's say, trimethylation event. Again, a different type of analysis. This was, remember before we did our study in a mouse model. Now we had all this patient data. Is there any connection? And before I argued that the mouse model is a very nice model for leukemia, recapitulates almost every feature of the disease, that we should be able to see in the molecular, if it's really true, we should be able to see it in the molecular level. We should be able to find some correspondence between the mouse genes and the human genes. So what we did here is kind of complicated, so you may want to pay close attention. So what we did is, on the one hand, we looked at the patient data, and we did all the correlations with all genes, with all these genes you see here. So take Jumanji expression across 600 patients and find which genes are co-expressed with that. Do the same for NF-κB1 and NF-κB2 with every other gene. Assume that this set of co-expressed genes is enriched in targets of that gene to some extent. I'm not saying all of them are going to be targets, but they should be enriched. And then go back to your mouse model where you found which genes have differences in K27. Which genes lost K27 when moving from normal double positive cells to leukemic cells? And ask the question, is there any enrichment? So is there any enrichment of Jumanji co-expressed genes in patients to genes that lost K27 in mouse? And you can really see this is really striking. You can see Jumanji is on top, UTX is not even close. Of course, EZH2 is a repressor, so it shouldn't be, so it's a nice negative control. And also a positive control is NF-κB, which Yannis' lab has shown to be activated by Notch. So all that was really convincing to us. Then we actually moved to do ChIP-seq of Jumanji in our Tcell, TLL cell lines, and we found that Jumanji is actually co-localized with not K27 trimethylation in leukemia, and not with K27 trimethylation, of course. And Panos did knockdown experiments to show that when you knock down Jumanji, but not UTX, you can see that the cells die in 2 different cell lines. And again, we want to go a bit deeper. We want to see how, at the molecular level, you can explain all this. And the first level is always to do an expression assay. So we did RNA-seq, and we saw that in leukemia, you have classic notch targets that go down. That's fine. But here, we need to show that Jumanji is important. So what does Jumanji do? Jumanji demethylates K27. So we need to go one step deeper. And we did ChIP-seq for K27 to really try to see if the mechanism is indeed, in this case, loss of K27 that can explain downregulation. And I think you can see that here. You can see the enrichment. You can see that genes that are downregulated when you knock down Jumanji. So you have a TLL, T-cell leukemia cell line, you knock down Jumanji, you know the cells are going to die. But why are they dying? Yes, because you're downloading these, you're downregulating these key genes. Yeah, too much data. Okay, so you're downregulating these key genes, but do you at least in general have a gain of K27? So does K27 play a role? And you can see that here. You can both see that genes that are downregulated are showing an increase in K27, and also as a negative control, I guess, you can see in green that some of the genes that go up, because some genes are going to go up, maybe apoptotic genes, they have the opposite effect, so decrease of K27. So it seems like K27, again, is playing a role. And remember, we knocked down Jumanji, which is a demethylase. So I think we're pretty good, because we showed downregulation of key genes. That's because of K27 increase, and we know that we affected Jumanji. And of course, when you do knockdown of UTX, we showed before that the cells don't die, so we don't expect any differences. Then again, in vivo, work done by Kevin Hu, actually, Ferrando Lab. And the next thing we did is, fine, so when you knock down UTX, you don't see any effect. So there's no point in going deeper. Can we do the opposite? Can we do actually something really dramatic, knock out UTX? So we did knockout mice, and we, again, tried to probe at the molecular level what's happening. Again, RNA-seq, these are different tumors. And personally, I was not very much convinced by this, because what we were trying to show here is fine. So when you have knockout, when you knock out UTX, you can see some classic tumor suppressors, or you can see some classic oncogenes. But you can always, the list of oncogenes and tumor suppressors is actually huge, so you can always find good examples. We found some pretty good examples in this case, but at that point, I was not convinced yet. But I can show you another slide later, so keep that in mind, that really convinced me that UTX is not just not oncogenic, but it's actually more of a tumor suppressive, has more of a tumor suppressive role. And remember that point I'm going to show you later. So the next thing we did is we used the Jumanji UTX inhibitor, K27 demethylase inhibitor by GSK that had just come out. So we tried it on different cell lines, and we showed that the cells die when you use that inhibitor. Again, we wanted to go one step deeper and really understand at the molecular level what's happening exactly. So we did RNA-seq and a bunch of ChIP-seqs. Again, you can see classic notch targets after several hours of treatment with the inhibitor. They actually go down. And again, our hypothesis is what? They go down because you affect the demethylases, particularly Jumanji, and then you have increase of K27 in those key genes. So the genes are downregulated, and so the cells die because the oncogenic program cannot be sustained anymore. And you can see that here. So you can see untreated, so before treatment, there is no K27. After treatment, you have increase of K27. Why? Because those are Jumanji targets, and of course you're inhibiting Jumanji. So if there's no demethylase, then EZH2, so that kind of answers Harris's question. So EZH2 has to be around so that it goes back and finds the opportunity to methylate again. So, and that's kind of what our not so good EZH2 ChIP-seq showed, but we just didn't trust it at that point to show it. But you can clearly see here that these genes downregulated when using the inhibitor are Jumanji targets. Some of them are not targets. They're all activated. You can see that. In leukemia, they don't have K27 in leukemia, but when you add the inhibitor, K27 comes back. And this, again, may be a bit complicated, but all I'm trying to do here is compare signatures. So we've done a bunch of RNA-seq experiments, comparing leukemia to normal in different ways. So here, for example, we did a knockdown of Jumanji. So in those kind of signatures, you have genes that go up and genes that go down. So that's why you need a matrix to compare every time, because you have the downregulated set and the upregulated set. So this first matrix shows the comparison of signatures of the Jumanji knockdown to the inhibitor. Okay, so what do we know from the phenotype? When you knock down Jumanji or when you apply the inhibitor, the cells die. So the signatures should be similar, and they are. So downregulated genes match downregulated genes, and upregulated genes match upregulated genes. So fine. So this, you can say, is an oncogene now. When you do the same for UTX knockdown, from the phenotype, we know that nothing happens. And you can see that. Not only that, you actually see the opposite. So you have a hint now that UTX can actually be not just doing nothing, but maybe have some flavor of a tumor suppressor. So you can actually see that the upregulated genes are the ones that match the downregulated genes. And the same exact story is when you go to the knockout. So again, I didn't trust the scatter plot before much, because you can always find good examples. But these statistics I trust more, because you can see on a global level that the signature of applying an inhibitor and killing the cells is kind of the opposite of the signature of having a knockout or knockdown of UTX. Too complicated? Okay, and going one step further, adding actually to the previous heat map I showed you. So the heat map looked impressive. Again, can you believe it? So it was a rapid increase of K27 trimethylation. Maybe, but maybe that's global. You know, you're affecting demethylases, and then okay, maybe EZH2 goes back and methylates everything. So you need I think at that point you need a control. You need to show that specifically Jumanji targets are the ones that are experiencing the highest levels of increase of K27. It's just that it's not global. It doesn't just happen everywhere. It just happens in the specific genes, and so you can actively implicate Genome-wide. And that's what you can see here. So you can see in a box plot, you can see the distribution of the fold changes of different classes of genes. The red class is Jumanji targets, and then the green class is that signature I mentioned before in the previous slide, genes that go down with the inhibitor but also go down when you knock down Jumanji. And you can see that both sets of genes have very high increase of K27, so you can link these two events. Whereas if you take another control, which is the opposite, genes that go up when you apply the inhibitor and knock down Jumanji, you can see the difference. Of course, it's statistically significant, but you can even see it's a big one. It's not just marginal. So in summary, and for all this we used an open source tool we have. It's free, of course. And yeah, I think I'm going to just skip those and acknowledge the people that did the work. Panos, the most amazing postdoc ever, did, I don't know, he probably did about 200 ChIP-seq RNA-seqs in total, some of them secretly from Yanni. And of course, Thomas also, Eva and Sophia did a lot of work. Sophia did experiments with the inhibitor millions of times. And from IBM, Tien and Filippo helped me with the analysis. From Ferrando Lab, and of course, Chibi, high performance computing dream team, Stratos, Eric, and Lauren. Thank you. Yes?
Did you try to find out why the Jumanji was upregulated in the cancer? Did you do any experiments looking at its promoter or the protein?
Did we? I think it's, we showed, yes, it's bound by NF-kappa B, right?
It's not an, oh yes, on that. It's an NF-kappa B target, NF-kappa B is high in this particular.
Did you look at any histone marks in the promoter?
We have them, but I think we just show the obvious. So we saw, of course, that it's bound, let's say, by NF-kappa B. We did not a ChIP-seq, but we did a ChIP for that. And yes, it was activated, so there was no K27 trimethylation, of course.
So do you have any explanation of why there's that peak, that thing going down with H3K27 tri in the tumor? Is that the nucleosome that's falling apart?
Could be.
Because
So
you see that with H3K4 tri, and
yeah.
you see the same thing go down because the nucleosome is open because it's
Exactly,
active transcription.
exactly. So one of the hypotheses was that the nucleosomes are evicted by knots somehow. I think we had some data for that, experimental, I mean. But yes, that's a very plausible explanation. And from our EZH2 experiment, a ChIP-seq experiment that I didn't show, you could actually see that EZH2 still binds there, not on the TSS though. So
CH2 is also phosphorylated, right? Did you look at has anyone looked at whether that's an inactive, it becomes phosphorylated, I think it's activated, or I'm not sure, I forget, activated, it's phosphorylated.
yeah, that's
through
in
signaling.
Yeah, so if there was some event that was preventing EZH2, yeah, could be. I don't know. So when we add the inhibitor, it seems like EZH2 can now work properly. So, but you can always argue that yes, Jumanji was doing the modification on EZH2 and couldn't do it, so now Jumanji is gone. So, but we never went that deep, I guess. Yes.
So, is anyone trying the inhibitor in human patients?
We don't know. We don't know. Do we know? Do we have any?
It's not bioavailable as it should be. So it is a lead compound, and the company is not particularly interested in following up because it's a very small disease.
Because it works in your mice, right?
It works with xenografts too.
So then when you say it's not bioavailable, I mean good
The
bio.
half-life is.
But you mean as KISH in general?
It works, but it's not for patients at this point.
Simply because they're not known.
Unless we find the Jumanji connection in a different disease that's maybe bigger, more important in that sense. Thank you.